All right, welcome to The Explainer. Today we're going to get into the truly ingenious science behind some of the most powerful and important medical tests ever developed.
So let's start with a huge challenge. Just think about this for a second. Our bodies are this incredibly complex soup of trillions upon trillions of different molecules. So how on earth can doctors find and measure a minuscule amount of just one specific thing, like a hormone or a drug or a virus?
Well, this brings us to what you could call the invisibility problem. And for the longest time, this was a massive roadblock in medicine. The early lab tests just weren't sensitive enough to find these key molecules, especially when they were present in such tiny, tiny amounts. For all intents and purposes, they were completely invisible.
So the big breakthrough, it was a total shift in thinking. The idea was, hey, if you can't see what you're looking for, don't just try to build a better microscope. Instead, what if you could attach a label to it, something you can easily see or detect? And that right there, that's the core idea behind a whole class of tests we call immunoassays. They basically use the body's own defense system, that really powerful, specific attraction between an antibody and its target antigen, and they combine it with some kind of measurable label. It's brilliant, really.
So how did they first crack this code? Well, let's dive into the first technology that really pulled this off, an idea so good it literally won a Nobel Prize and changed medicine forever.
So back in 1960, this brand new technique comes along called radioimmunoassay, or RIA for short. It was originally developed to measure insulin in the blood. Now this thing was so groundbreaking, it completely changed the game for research and diagnostics, so much so that its co-creator, Rosalind Yalow, snagged a Nobel Prize for it.
Okay, so what was this clever label they used? Well, as the name kind of gives away, RIA used a tiny but very easy to detect radioactive tag. Usually it was a radioisotope of iodine, and you could measure it with a special instrument called a gamma counter.
Okay, here's where the real genius kicks in. Because it's one thing to tag something, right? But how does measuring that radioactive tag actually tell you how much of a completely different, unlabeled substance is in a patient's blood?
Well, the way it works is honestly like a game of molecular musical chairs. It's amazing. Just imagine the antibodies. Those are the things that are going to grab onto the substance we're trying to measure. Imagine they're just a limited number of chairs.
So here's what we do in the lab. We have two groups of players. We take a known amount of our radiolabeled molecules, let's call them the red shirt players, and we mix them in with the patient sample. Now that sample has the unlabeled molecules we want to measure. Those are the blue shirt players. Now you've got both teams competing for the same limited number of chairs.
Okay, so here's the crucial part. If your patient has a ton of the substance, a whole lot of blue shirt players, well, they're going to grab most of the antibody chairs. That leaves very few spots for our radioactive red shirt players, which means the radioactive signal is low. But if the patient has very little of the substance, there are plenty of empty chairs. So more of our labeled molecules get a seat, and the signal is high.
And just like that, by measuring something we can easily see, that radioactive signal, we can precisely calculate the amount of the thing we couldn't see. It's this super clever, indirect way of getting an exact measurement. So RIA was a total revolution, no question. But, working with radioactive materials has its challenges. You've got safety concerns, disposal issues, you need special equipment. And all of this got scientists thinking and asking a really important question. And that's the exact question that drove the next big evolution in diagnostics. Could we get that same incredible power and sensitivity that RIA had, but, in a safer, cheaper, and more accessible package?
And the answer was yes. Enter ELISA. So with the ELISA, instead of a radioactive tag, we use an enzyme as the label. You can think of this enzyme as like a tiny little factory that we've attached to our antibody, and it's just waiting to be switched on.
Here's how it works. Once the antibody with its little enzyme factory has found and bound to its target, we add a specific chemical called a substrate. Now this substrate is colorless at first, but when it comes into contact with that enzyme factory, bam! The factory switches on and starts churning out product that has a very visible color. So, remember how with RIA, more substance meant less signal? Well, with the most common type of ELISA, it's the total opposite. It's a direct relationship. The more substance there is in the sample, the more of those enzyme factories are present, and the more intense the color change becomes. Simple, visual, and incredibly effective.
And this table just lays out the evolution perfectly. With ELISA, you're swapping a radioactive isotope for a totally safe enzyme. You're swapping a big, complicated gamma counter for a simple scanner that just measures color. I mean, this upgrade made the whole process cheaper, safer, and way, way easier to perform, which is why it became a cornerstone of pretty much every modern lab.
Okay, so what's the big takeaway here? Why does any of this matter to you? It's because these techniques, they're not just some abstract science. They are the absolute engine behind modern diagnostics.
I mean, think about it. From a home pregnancy test that you can buy at the drugstore, to the super-sensitive tests that diagnose infectious diseases like HIV, to just a routine check of your hormone levels at the doctor's office, the principles of these immunoassays are working behind the scenes in all of them.
Let's just circle back for a second and appreciate how mind-blowingly sensitive these tests really are. The original RIA could detect a substance at the picogram level. A picogram, that's 1/1,000,000th of a gram.
What does that even mean? Well, to put it in perspective, that is like trying to find one single specific grain of sand on a beach that is a mile long. That level of precision, it's almost impossible to wrap your head around. That is what allowed scientists, for the very first time, to accurately measure things like hormones, completely changing our understanding of medicine.
So when you think about it, we've gone from a radioactive tag that a special machine can see to a tiny enzyme factory that literally paints us a picture with color. These really simple but absolutely brilliant ideas of labeling the invisible, well, they've saved countless lives, and they continue to push medicine forward every single day. And it really makes you wonder, right? What comes next? What's going to be the next ingenious label that changes everything all over again?